And now, before the roundtable, we're going to be hearing from Adina, Michael-Titus, with the right title, Omega-3 fatty acids and spinal cord injuries and traumatic brain injuries.
Thank you. And first of all, thank you, Larry, for the invitation to attend this exciting symposium. Thank you, Richard, for suggesting we share some of our results on omega-3 fatty acids in the treatment of neurological injuries. I'm a neuroscientist and a pharmacologist, and I lead the Center for Neuroscience, Surgery and Trauma at Barts and the Royal London School of Medicine in London. And we are associated with a very large level 1 trauma center, one of the largest in Europe. So we have a lot of interest in neurotrauma, in particular traumatic brain injury and traumatic spinal cord injury. And this montage of images illustrates how this condition arises, whether it's in the civilian sector or the military sector. And they are conditions which often can lead to life-changing consequences. Traumatic brain injury, for example, is the leading cause of mortality and morbidity all over the world in younger people. But the demographics around the world is changing. And we can see now that there are many more older people affected by these injuries. And last but not least, there is now clear evidence that even experiencing a moderate traumatic brain injury increases significantly the risk of developing neurodegeneration later. So what is a timeline of the care pathway when an individual has an injury? We have here an image of an emergency team, trauma team. It could be somebody flying with a helicopter or a car service, and the casualty is transferred to the trauma unit, and there will be also later on a transfer to the intensive care services, and then at some point the patient is discharged. And from the therapeutics point of view, we think of the first acute, subacute phase, the first minute, hours, days after injury, as a neuroprotection space when we should protect the injured brain and spinal cord. And later on, in the protracted phase after injury, through neurorehabilitation, for example, try and promote some function again, and hopefully assist with neuro repair. So now you would expect the next slide to be an example of what we have in terms of treatments, whether it's neuroprotection and neuro repair in this area. And you would be disappointed because the next slide would be blank, because we don't have anything. So if you would want to open a book on pharmacology and therapeutics and read about how we treat in 2024 traumatic brain injury and spinal cord injury, I'm afraid there is nothing. So the challenge is immense. So just to give an example of, now going to the detail of mechanisms, example of what happens when, for example, there is an injury at cervical level, which is a very common level of injury in patients. And after the injury occurs, there is a point at the epicenter of the injury where the tissue is lost irreversibly through neuronal cell loss, also non-neuronal cells like the glia, astrocyte, microglia. So there is a zone of destruction that you can't reverse. But after that, what happens is that this injury zone expands, and we call this the secondary injury phase. And unfortunately, this expansion has damaging consequences. There is intense activation of a neuroinflammatory response, and there is also formation of a kind of scar, a wall of cells around the injured side. Initially, this wall of cells has a protective role, but it can become a very non-permissive environment for regrowth and for any attempt the tissue may have to repair itself. And indeed, in the tissue, there are some mechanisms. For example, you see here some axons which try to extend neurites, new growth, to repair the disrupted connectivity of the cord. But this is very limited, unfortunately. And the presence of this scar, which is very intense as time goes by, makes this environment very hostile. And ultimately, what we see is inside the spinal cord, whether it's experimental animals or patients, cavitation zones, sometimes filled with fluid. So if we look at a real case and talk about this secondary injury and its evolution, here you have the example of a young boy, a motorcycle accident. And he had, again, a high cervical impact. And if you follow the injury during imaging, you see that the expansion of the abnormal zone is very rapid. In less than 2 days after the injury, you have an expansion of the abnormal signal from around 7 centimeters to 10 centimeters. And this expansion is really bad news because it means that you have you affect a far larger amount of tissue. And this is why neuroprotection strategies target this concept of secondary injury and how to prevent the spread of injury. So if you want to look at the biochemistry of the pathophysiology, what happens after the traumatic spinal cord injury, and to a large extent, there are many similarities with also traumatic brain injury. There is an avalanche of biochemical processes which are time sensitive. Initially, there is a lot of necrosis, there is a lot of excitotoxicity, a lot of glutamate is released in the tissue, which activates receptors and leads to destruction in the perilesional zone. There is intense oxidation of proteins, of lipids, of nucleic acids, and also an inflammatory reaction, a very complex inflammatory reaction that was referred to by Rouron earlier in the case of injury. This is a very strong endogenous response to injury, either through local cells, microglia, astrocytes, or through cells that invade the injured tissue from the periphery. And there is also a lot of axonal distress and axonal damage. So when you look at this, you may think, oh, there's plenty of scope to intervene. And why don't we have anything? Why do we have a blank page? And there is a famous cartoon in Nature 2008 or so, I think, talking about this valley of death, trying to develop something which works in the clinic. It works in animals, and then you want to move to the clinic, and there's a lot of loss of lives. Mostly, I think, it's scientists' lives are supposed to be in this valley of death. And lessons have been learned because millions, billions have been spent in the last 2 decades through failed attempts to move something which looked good in animals to move successfully to the clinic. And we've learned that it needs to be a very thorough investigation, and some important questions need to be addressed. It needs to be clear evidence of efficacy across the models of injury, because a unique characteristic of this condition in humans is extreme heterogeneity. You may remember that Hippocrates said, there is no such thing as disease, there are only patients. And this is a unique condition in medicine where indeed it's an n equals 1. So you need to make sure that you don't choose your favorite model of injury and then you prove something phenomenal and then you're going to move very fast to the clinic. And there are some other pragmatic aspects which are summarized here, linked to defining dose, regime of administration, time window, and so on. And time is tissue after neurological injury. So all these things need to be thought through. And the fact that they haven't been thought through, it's very easy to make omissions, explains at least partly the rate of failure we've had in this field. So now I would like to move to this compound, very interesting compound mentioned already by Richard, am I wrong? DHA, a long-chain omega-3 fatty acids. And first, explore with you some of the data in traumatic spinal cord injury. As already explained, we all know that DHA is very enriched in the brain, very important for development of the brain. But apart from this, there are two other characteristics. Yes, it is in membranes, it's there, it's structural, but also acts as a ligand for a variety of receptors, GPCRs, transcription factors, voltage, gated channels, for example, and produces, of course, this hugely complex family of SPMs that Rurong has explained so well a moment ago, and they have their own signaling system. So a very complex molecule. So for us, how did it all begin? I read this paper on linolenic acid being injected after transient spinal cord ischemic episode. This was work done in France by Michel Lasdunsky and Catherine Hortot many years, more than 20 years ago. And it was just a transient ischemia, but they were showing very clearly, looking at neurons in the cord, that they looked much better after they injected this fatty acid, which is the precursor to DHA, as Richard explained. They injected it intravenously at a ridiculously small dose, around 0.1 milligrams or so. And at the time, we were working on DHA, and we were very aware that there are similarities between linolenic acid and DHA. Linolenic acid is a ligand for the TREC1 potassium channel. And this is an interesting target because it responds to mechanical stretch. So DHA, like linolenic acid, responds to mechanical stretch, probably through TREC1 targeting. And you can see here that TREC1 can be expressed in the presynaptic zone and may control, for example, the release of excitatory amino acids. And this is a very important concept. And it's clear that it's present in the adult central nervous system. You see here some example of, through immunohistochemistry, example of distribution of this channel in the human tissue. So we hypothesized that apart from this transient spinal cord ischemia, there may be another story which would be much more important to test whether it can help in traumatic injury of the tissue. And this is what we did. So we took a variety of fatty acids, including linolenic acid and DHA, and we did first a hemisection type of injury in the rat, the thoracic level. So imagine that this is the equivalent of a knife stabbing. Sadly, in the place where I'm coming from, in London, we have a lot of those. So you actually cut the cord, and you can see there is a zone of injury, which is very clear around the cord. And when we injected DHA in these various fatty acids, we clearly saw that DHA injected intravenously reduces the lesion incredibly. And other fatty acids, such as arachidonic acid, which, as Richard alluded to, is a pro-inflammatory agent overall, led to a much worse aspect of the tissue. And we looked at protection of neurons, counted neurons in the injured tissue, and there was clear protection after DHA, this unique bolus injection, very low dose. And we looked at a multitude of parameters. I won't have time to explain, but you can do various types of scores, for example, BBB score, which looks at motor performance, foot slips on a ladder. And DHA was definitely very active, and linolenic acid also had some activity. But this is not enough. And as I said a moment ago, in the field, there is now a lot of skepticism when you come out with your first study. You may publish it in the end, but it's not enough. So we move then to compression injury, which is the equivalent of a very strong compressive force, which looks terrible after weeks of after injury. In animals, you see this extreme cavitation in the cord. And we looked at various parameters, for example, the inflammatory response. And when we injected DHA, and this time we had a group receiving a single bolus intravenously of DHA, but also a group where we had DHA as an injection, and we enriched the diet of the animals with DHA also over several weeks to see if we could amplify the effect. And we saw a very clear decreased neuroinflammatory response. We saw a lot of protection of key cells, such as the oligodendrocytes that Rurong alluded to, very important for myelin. And in some cases, as you can see here, for some markers, having the diet helped a little bit more. But definitely, the driving force of the effect was this injection of the DHA bolus. So also, we looked in detail of the axonal distress induced by injury. Axons are so important for connectivity, whether it's the spinal cord or the brain. And axons transport proteins, and we can see that after injury, there is an accumulation of proteins. You see these blobs, fluorescent accumulations. Proteins can't be transported. When you injected DHA, everything was clean. Look at this beautiful cord. We couldn't see anything. And if you look at the structure of axons with a myelin sheath in green, and in the red signal, you see the middle of the axon through neurofilament staining. Injury creates a state of complete chaos in tissue, destruction of the tissue. And when we treated with DHA, this was much improved. So, not enough. We move then to cervical level injury, which is very common in patients. Here we can test for impairment in the forelimb function of rodents. And as you can see, once you injure a rodent, there is no ability to grasp pellets, for example, to eat. You can test both for motor function, which is crude, or finer motor function, the ability to move the component elements of the paw, the digits. And we saw that after injecting DHA, there was some improvement in performance in this type of injury. So clearly, it was segment independent. We had a protection which was clear. And in this model at cervical level, we also looked at this very interesting response of the body, the sprouting of axons. And you may be able to see, we quantified it, but you may be able to see just about through immunofluorescence, there are more sprouts when you inject this bolus of DHA than without DHA. So DHA seems to enhance the intrinsic plasticity after this type of severe injury at cervical level, which leads to significant improvement of the forelimb, impairment in the function of the forelimb. And also, we looked at various synaptic markers. You see these beautiful red cells, the motor neurons. We showed that the synapses and the drivers of their activity, serotonergic innervation, were much improved after DHA. And you will say, okay, so what about the second species? It's rat, but we did some work in mice, and we published an extensive study side by side in the most severe injury it can induce, which is contusive injury. This contusion injury can be induced by letting an impactor drop on the cord, and you create a lot of damage. But it's very realistic, because think of earthquakes around the world and situations where there are also buildings that collapse. And you can have elements of significant weight dropping on the cord. And here, without going into detail, we saw much better recovery with DHA, exactly with the same approach. And also, we did some mechanistic work. Rurong alluded to microglia, and they are very important in the inflammatory reaction. They are very keen cells to come and help with the healing after injury, but they can go in the kind of state into overdrive, if you wanted to, and you end up having too much of a good thing, and microglia eating too much myelin, for example, in the zone of injury. And what we've discovered doing imaging of myelin inside macrophages and microglia, we discovered that DHA refines this response and modulates this response, which was really very nice to see. I would like to touch very briefly on the aspect of control of complications. As beautifully described in the talk this morning early, neuropathic pain is a concern after spinal cord injury, and rats do develop it in a similar manner to humans. And the question is, does DHA affect this particular aspect? And this is work done by somebody who worked with me and then created his group in Scotland, Wen Long Huang, and his students. And he's published this study. I'm going to take you just very briefly through the protocol. This time, it's not only a single bolus, 30 minutes intravenously after injury. He gave because neuropathic pain takes time to develop. I think there was a question earlier today, what is the window, the time window when this plasticity, which sometimes abnormal plasticity, develops after an injury? And in rodents, too, it takes time. So Wen Long decided to continue injecting every few days over weeks. And then he explored this phenomena of hypersensitivity of the animal. And you can see here, for example, a decreased response to what we expect to be a normal paw withdrawal response. This is a consequence of injury. The animals are hypersensitive. When you inject DHA and you start in the acute phase, you can alter this type of response, the paw withdrawal response. So you affect a component of this abnormal sensory perception. And interestingly enough, he showed that there was some effect which was present even if he delayed the first bolus of DHA a few weeks after injury. So this is this other graph. So there was something possible even if you missed the first phase. And of course, the question is, how does this work? And so he did more analysis of the behavior of the animals, apart from the simple paw withdrawal assessment, which is standard assessment. And he showed that indeed the animals have profound behavioral abnormalities. They would scan an open field, for example, very anxious, wouldn't go into the center, and DHA would reverse this. And in terms of molecular analysis, cellular analysis, what happened, essentially it's all about microglia. Again, microglia go into hyperactivation, and you can stain them with specific markers which show the state of hyperactivation. And when you inject DHA, you see a very clear reduction. You can quantitate these microglia which are activated in the tissue, and you see that this is probably a relevant correlate of the effect of DHA. And you can also measure the levels of pro-inflammatory cytokines in the tissue and also in the dorsal root ganglia involved in the development of neuropathic pain. And you see that there is a very clear indication that DHA across the board, so key pro-inflammatory responses, they are dampened by this bolus of DHA and repeated over weeks. And he did a test in parallel with pregabalin in some of the animals who had neuropathic pain. And he showed that if you compare the intensity of the dampening of this neuropathic response, it's very comparable in DHA and in animals treated with pregabalin, which is very important because pregabalin can be used in patients. It's a good term of reference. Last but not least, I want to mention the fact that we also looked at what happens if in animals, like in people, you rehabilitate animals after injury, which you do in patients. Does DHA offer an additional advantage? Is there a synergism? And as you can see here in the molar cervical injury, we showed that there was clear synergism of action. And the optimum results, again, here we're looking at the fine motor function recovery. The best results you can see when you have a neuroprotectant in the acute phase and then rehabilitation. So now I will like to move very briefly to some evidence in traumatic brain injury and focusing just on 2 or 3 examples as we lack time for more in-depth analysis. we and other people have shown efficacy of this bolus approach, intravenous DHA, in models of injury. Here, we are looking at a model of contusive, unilateral, contusive open skull injury, which is quite an injury for rodents. And again, just to guide you a little bit through these graphs, you can see that we have a score of injury, which is an integrated motor sensory impairment score, which is very high after injury. The animals are impaired compared to sham surgery animals in pale blue. And DHA injected as a bolus reduces this impairment. And when you look in the spinal cord, again, we've done an extensive analysis. For example, if you look at microglia, they become hyperactivated, ameboid after injury, and dampened after the bolus of DHA. And more importantly, we showed that you can use a biomarker which is very relevant in a clinic. In patients with traumatic brain injury or spinal cord injury, you can measure neurofilament L, which is a structural component of axons. It goes high after injury in the cerebrospinal fluid and also in plasma. And you can use the same marker in rodents, and you can see DHA reduces this increase in neurofilament L, which is very objective proof that something meaningful happens in the central compartment in terms of protection. And in collaboration with a colleague who measured for us lipid mediators, indeed, we showed that there is a shift to pro-resolving mediators after an injection of DHA. And just to give you another example, now we have data from various species and studies with various models in different species. I haven't said much about impairment of the blood-brain barrier and also impairment of the microvasculature, which happens after injury. And here you see, again, a very clear impairment. Access of immunoglobulins is enhanced after injury, so there is a destroyed blood-brain barrier, and DHA improves this aspect. And finally, also, there is a loss of microvessels, you see here, and DHA improves this aspect too. This is a model, again, of invasive injury in the rat. And all this occurs in parallel with an improvement of a neurological nature in animals. So, we have come a long way. I have memories of sending our paper to a very respectable journal when we first saw the effect of DHA bolus in spinal cord injury. And we received the kind of letter and feedback from reviewers and editor you don't want to receive very often in your career as a scientist. I think I should have framed it. I should have kept it in a nice frame. But somebody quoted Churchill earlier today, never, never give up. And we continued, and the field has moved to the acceptance that this is a very interesting story. There are studies from various groups, and you are going to see in a corner study from the Deckelbaum group, remarkable package of studies around DHA in the model of neonatal encephalopathy Richard is interested in. So we have come a long way, and it was true, and it is true that this is a very interesting story. This is a fascinating lipid story, and I hope that it will help us make a difference to the quality of life of patients. And I hope we can move now to the clinic with confidence that perhaps we have a chance to indeed neuroprotect in the early phase and make a difference in the long term. So nothing is possible without wonderful people, and I owe a huge debt of gratitude to many colleagues. Special place in my heart to my late colleague, Professor John Priestley, who was my companion on this scientific journey at the beginning, and all the others who have worked with me and have believed in this story, which has become now a worldwide accepted story. The lipid research is fascinating, as also Rouron explained earlier with the SBM story. And I think there is an SBM story and there is a DHA story. And there is a new chapter of therapeutics, which I'm very excited about, because indeed it is a chapter focused on lipids, and this is new. And in my view, I think there are barriers of an objective kind when we don't know enough, but there are also barriers in the mind that we need to overcome to believe that the impossible is possible. And yes, lipids are structural and have a variety of roles, and they can do things which are normally we don't associate with lipids at all. But they might have the answer, at least I hope I do, to some of our most complex questions in neurotherapeutics for which we have no answers at present. So thank you again for having me today.
Great. Well, I stay here. So before we go onto the round table, we're going to have some specific questions for Adina. And Larry, looks like you're the closest that I see with the hand up.
I would like to ask a question that everyone wanted to ask, I think. I mean, I only do that. I mean, so could you go back, if you still have your slides, to that picture of the hemi section, the first one you showed us, where the...
You need to go back. I think you're going forward into my reserve slides. I had many of those. You need to go back.
Further, back.
Probably better to go to the beginning, probably. It's better. Yeah, it's what was in the first part of the
It was
talk.
about the third or fourth or
Yeah.
fifth slide or something. Keep going. I know the slide.
It's coming.
Gonna never forget it.
We didn't forget it either, because we repeated those experiments, Larry, because we almost couldn't believe it. I think you've missed it.
I think you just missed it. Yep.
You've missed it. You need to,
It's really hard to believe we missed it.
You need to
very
go
quickly.
back.
And the thing that
Yeah.
happened when you showed that slide,
Yes.
this was I mean, I don't know this model, but there was a cut made.
Yes, there is a cut.
A cut made, and then, although there was not lots of data, there was a slide, a cut, and then a heel.
Yeah.
And again, everyone started thinking about stem cells and regrowth and no paralysis
Yes.
and all that. So what is the connection between the way you're thinking about DHA
Yes.
and this idea that somehow people who have suffered from spinal cord injuries could end up walking again because of the apparent lack of injury or the growth, whatever that slide showed? You must think about that all the time.
Yeah, I do think about it. And thank you for asking the question, because first of all, when we saw it, Larry, it may sound ridiculous to say it because we had a hypothesis, so we should have been pleased. We were worried that it looked so incredible, that the lesion was almost, the parallel edges were so close. And we thought we must have done something silly. And we repeated that twice. That's why I didn't particularly appreciate the comments made when we sent this for publication. But it was true. And now, is it possible to have total healing in spinal cord injury? I think this is a holy grail. Hemisection, I would agree with this idea that hemisection is a kind injury because it's a very neat cut. And we know already there is an astrocytic and glial reaction. It's nothing like the real thing, which is more compressive and contusive and complicated by the distraction, dislocation forces, and so on. And there is a lot of interest in using cells locally, for example, stem cells. And nothing has come out of those studies yet. I do not think that you can have, through DHA only, a complete miraculous restoration of function. And I think one needs to be also very aware that we still don't understand when we see recovery in quadrupeds. They have a central pattern locomotor generator in the spine, and they recover much better than non-rodents and quadrupeds. We are bipeds and we pay a price for it. And we also pay a price for having a lot of white matter compared to a rodent. So I think the DHA, or whatever you work on that you believe may have a neuroprotective potential, may reduce the injury. But some injuries may be of such an intensity as severity that it will be unrealistic, I think, to just give a bolus of DHA. I think the spinal cord will be repaired. Perhaps the discussion will have, I'm very aware of the time, I can tell you about some other studies we are doing, because DHA, we mustn't forget, has a structural role in the formation of phospholipids, and you can also support that structural aspect of the importance of omega-3s. But I would like to go back to this concept of use, saying, well, the hope of people is that they are going to walk again. You may have seen in journals like Nature reports from the Grégoire Courtine lab in Zurich, and they have managed to take chronic injured patients and with stimulators, and there's some very nice work which was done initially in the States by Suzy Arkema, but Greg Courtine is doing it in Zurich. Stimulators, very complex patterns of stimulator, a kind of stimulator pad, because you can have, based on proprioceptive information, you can, it's a very clever electronics, and you can have a feedback system which allows people to start ambulating. It's a very complex device, and it's still experimental, but there are some very spectacular effects. And it must be, it must be said that in the field of spinal cord injury in particular, in traumatic brain injury, it's even messier. Let's not go there. But in spinal cord injury, some people think, well, neuroprotection in spinal cord injury is so complicated, just forget it. Give up this concept. Just focus on the chronic phase and then try and put some stem cells, try and put some stimulators in, and so on. So we'll have to see where we go in the next decade.
And certainly one thing that hasn't been very studied is, or studied well, what's the interaction of stem cells and omega-3s? Because a big part of the omega-3s is not just stopping the tissues from dying or getting hurt, but they're also very important in regeneration. We've heard some of it this morning, but not a lot, and there's a big role for omega-3s in regeneration.
Yes, DHA enhances neurogenesis, absolutely right, yeah, which is another compensatory mechanism when you lose cells, but it's not enough. You can have cell proliferation perilesionally and also in the typical niches in the brain where you have this formation of neural precursors. But this may be helpful, for example, in normal aging, but after injury, it's probably not sufficient. I'm talking too much.